And now let's turn to Mr. Debasish Sahoo, who is with us, also from India. And I cut his time a little bit short, so I will make it up at the end of the presentation, of course, yeah, because after that we've got coffee break. So of course I will give you time to speak a little bit longer. So thank you, Dr. Natarajan. And now let me ask Mr. Debashish Sahu to give us his presentation, Research Approach and Commercial Potential of Herbal-Based Therapy Using Modern Technology. Please, Mr. Sahu, you can speak now.
Thank you very much for this opportunity. Some of the most important part is that today herbal drug system is going to be the next generation drug system, I think. So for that, we need many advancement in the research area, some approaches from our side, from the different research communities who are into the herbal drug system, who are developing rapidly the drug system into a modern type of systems. And we can increase the commercial potential using the modern technologies that we are, and some of the concepts and prospect of ours from our side, and we can enhance the safety, reliability, and authenticity of that herbal drug system, along with the most important part is the conservation of the natural resources that we have, that relies on which the herbal drug system totally relies. So my first presentation, the first slide will describe you regarding some introduction. So herbal-based drug systems are used to treat, cure, enhance healthiness of the mankind. Okay, so it is by the ethnobotanical prospect and application. So these drug systems have used in many ancient cultures. Okay, so many drug systems that are ancient cultures are Ayurvedic, Chinese. So in some Japanese cultures, there are also medicine systems. Some Korean medicine systems are also there, which uses the natural resources, the plant materials, and their drug abilities in their medical system or healthcare system. So naturally, these natural products have some chemical compounds that can use to treat some diseases, and these chemical compounds can be enhanced in a much more technical and much more applied technologies where we can find out and we can discover the new drugs. So with the advancement of the drug discovery process, different toxicological studies, drug-drug interactions, many theoretical knowledges and therapeutic principles are there. And we can explore those principles, the modern pharmacological and mode of actions by use of different advanced techniques, along with systemic and ethnobotanical traditional medicine, can make this system more understandable in a more precise manner. So my main objective is to find out, conceptualize, and scientific approach leading to modern and high throughput drug discovery process associated with herbal drug system, overcoming challenges that renders better acceptability in modern world and globalization of local and ethnopharmacological knowledges. So first we have to understand what is a herbal drug. So the plant material, they constitute the chemical compounds. Those are called the bioactive compounds. Those bioactive compounds are having different effects when we take a herbal drug or herbal component. So it has a different effect. A single drug can target different types of other means diseases. So on an whole, it is called as multi-drug, multi-target concept. So it is a type of combinational therapy that is found to be very, very effective and very, very efficient. Okay. So this network pharmacological data and big data can help us to find out the possibility where we can correlate the synergy and other mechanism for developing the prospect of the natural products. So different approaches, some of the approaches that I want to describe in this system. One is sustainable agriculture practice of herbal raw material. As we know, in the APIs or chemical compounds, we can synthesize in a chemical means combinational chemistry. But for a herbal raw material, we have to rely on the plant material. So those plant materials that contains different types of phytoconstituents, so such as alkaloids, phenolic, saponin, tannin, amino acids, carbohydrate, terpenoids, and flavonoids. So this chemical compound can function alone. Aluchas, in the conjugation with the phytochemicals, they produce different pharmacological effects. So as a whole, these types of this drug system that relies on the herbal drug material has to be practiced on a sustainable agricultural practice where we get an authenticate raw material and authentic and a good quality raw material so that the drug system will be having a more efficient and more effective way. The second one is called good handling practice, good manufacturing practice, and good distribution practice. With the help of good agricultural practice also, there are also different types of other practices that need to be addressed in a very scientific manner. Whenever we are handling those different types of herbal materials, good handling practices, as well as wherever the manufacturing is going on, good manufacturing practices should be there, as well as good distribution practices should be there. So these all these things post-harvest conditions and handling, such as drying, processing, transportation, storage, extraction, has a major impact, and it will deteriorate the quality, quantity, and efficiency of the batches, and is also it also helps in the variation of the batches. So what happens if there will be variation in the batches? Then your the herbal drug system will cannot we cannot rely 100% on just particular. So another thing is that contaminations. Different types of contaminations with the means the raw material, such as the heavy metals, the pesticide, insecticide. They have also a deteriorated effect on the drug system because these heavy metals and these have as well as the type of pesticide and insecticide. They have different types of toxicity, such as hepatotoxicity, cardiotoxicity, neurotoxicity, and hematotoxicity, as well as there for a good handling practices and good agricultural practice, we have to analyze each and every raw material. At least we can do the quality control by estimating by different by estimations with different analytical techniques such as SPLC, AAS, and mass spectroscopy. So process attributes and quality control. So extraction of active compounds plays a major role. So different types of extraction methodologies are there. So you have to optimize the methodologies, whether we'll go for cold extraction, whether we'll go for hot extraction, whether we'll go for decoction or maceration. So it depends basically most importantly on the pharmacological effect, which extraction process has the maximum pharmacological intended maximum pharmacological effect. Secondly, there are also proper research and observations should be there based upon the stability of the raw material, stability of the intermediates, and stability of the finished products, as well as the whole time study, process attributes such as milling, sieving, blending, granulation, compression, block density. These also plays an important role in maintaining the integrity of the drug system. So with the increase in the shelf life, better theoretical ability, better research on in vitro and in vivo stabilization, better absorption, better bioavailability, stability of raw material, reduce negative interaction and limited toxicity. Then pharmacogenomic approach. Pharmacogenomic approach right now what happens is that whenever you take a drug, a doctor prescribes a single drug to many patients. So that drug can have an effect with 10 tablets, can have effect on 100 tablets. So it depends upon the genomic integration of the different individuals. So right now, if we integrate this pharmacogenomic approach where the different genetic makeup of the organism or the individuals can be integrated with the drug system or integrated into the pharmacological study, we can make the drug system more effective. We can analyze the qualitative and quantitative the discrimination in between the individuals having the different genomic in based upon the different races, based upon the different countries. So this development in the genetic, proteomic, and metamolecular tool will fulfill the study of therapeutic interaction, bioavailability of the drug compounds. So integration of this public health with the study of the individual genomic, genetic, and physiological aspect can render numerous study profile for better therapy. However, efficiency of the drug metabolism depend on the presence of drug metabolizing enzymes. Okay, so our near future will be on the futuristic medicine or the more personalized diagnostic with the help of different diagnostic system and help of different, I think with the help of different AI and other tools, we can integrate this pharmacogenomic more efficiently into the therapeutic potential of the drug system. So, role of information technology, computational biology, and data mining. As of you all know, that bioinformatics and computational biology are playing a pivotal role in the development of drugs. So, right, like that, also, if we have information technology, if we have integrated database for Ayurvedic system, for Ayurvedic practice, and Ayurvedic outcomes. For example, for example, there should be a database for herbal medication. There should be databases for the outcome of the different types of diseases. There should be type of medications and their outcome and their correlation with the qualitative and quantitative integrations. So these different types of things that the in the information technology. So these types of databases to the chemical fingerprinting as well as the cytokine composition can be correlated with the different efficiency of the drug compound. So these databases can also give an insight to this quantitative user and quantitative composition activity relations. So virtual screening approach can also be used for plant-based drug system by using available online libraries for bio and plant metabolites. Another system is the reverse pharmacological approach for drug discovery process. As of you know, in the Ayurvedic system, it is already been established. Many Ayurvedic practitioners are there. Those who are practicing the Ayurvedic system from the generation to generation. So they know a much amount. We have by much amount of data regarding the pharmacological, regarding the therapeutical value of this drug system. So once we gather this information from those things and integrate into the machine learning system, so we can get into new drug system, new development of the drug system, where we can reduce the amount of time. We can reduce the amount of time for development of the new drug. So this is called reverse pharmacological approach. So you can see whenever there is a development of a new drug, first we have to identify a disease, then we identify a protein, then we go for development of different types of target lead identification. Then we can go for lead optimization. In lead identification, there will be about 1 lakh compounds. In lead optimization, there will be about 1,000 compounds. Then we'll go for preclinical trials upon animal trial, upon human trial. Then at last there will be a drug system. But in a reverse one, what happens? So whatever safety studies are there, whatever the pharmacological studies are there, whatever the toxicological studies are there, they have been already been seen by the efficient and more prominent Ayurvedic practitioner, more prominent doctors, those who are into the field from generation to generations. So from their data, from their experimental studies, from their drug, from their doses and doses concentration, whatever they are scheduling to the patients, along with that, whatever they see of the lifestyle of the persons, the therapeutic and toxicological parameter. So this can get integrated into different modern drug designing system to get into an efficient, safety, and stable drug composition. So artificial and machine learning in herbal drug system development and authentication process is a very important part. So the process of drug discovery and drug development is a very complex technology and is multi-component strategy. So these synergetic combinations can only be possible by network-based system. As I have told earlier that we should develop a database system. We should develop some type of integration networking system where we can integrate all those data into a single piece, into a single world. So that whenever one practitioner wants to get some data about the patient, about the regime, about the different level of drug, what the combinations of the drug, and quantity of the drug. So they can easily find out, okay, this is the regime that has given to a patient. So we can try out a new system or we can try out a new regime. We can experiment a new regime on different types of regimes. So like that, we'll get the experience and we'll get all the things, how to accumulate on different types of different elements such as liver diseases, cancers, diabetes, etc. So study of entropy, herbal biomolecule disease network system that will depict the synergistic effect of combinational drug compound. Machine learning technique can also be used to build up QSAR and QCAR. Web-based and mobile application can be used for the recognition of the medicinal plant. For example, I am an individual. I have gone to a park, I have gone to a herbal park. So I need to see what is the type of plant, what is the use of the plant. So simple by clicking on a mobile, and if I have a technology, if I have a database, simple by the click of a system, I can find out just like a Google Lens. Just I can find out what is the plant, what is the utility of the plant, how the plant is useful in different elements. Okay. So automatically this will help in recognition of the different plants. Along with it will help the local population, different taxonomists and biologists to provide proper identification and authentication of the plant species and plant material, along with increased knowledge of plant material. So this all will be helpful whenever we have a proper data systems. This will also help in the conservation of local flora. Okay. So integration of DNA fingerprinting, DNA barcoding can also integrated with the artificial intelligence to make more authentication more reliable. Then biotechnological prospect. So DNA barcoding is the identification of the plant material for the drug isolation and compounding. So DNA barcoding method is a short sequence of DNA from single cell of an organism that differentiates the species from other in the same kingdom. This has served to be the better and standard method for species identification that matches the DNA barcode sequence from the unidentified organism and match the DNA barcode sequence with comprehensive libraries of plant barcode of live data. So then DNA-based molecular markers. These are also molecular markers that help in identification, that helps in finding out what is the differences between the two plant species, how they have evolved through the evolution. Chemical fingerprinting is also can be also integrated into biological fingerprinting, where chemical fingerprinting means we have a single type of molecules that is present on a single plant. So the quantity and quality of the particular type of chemical in that chemical molecule in that plant especially can authenticate us that this is the plant and this can be the parent plant or a native plant. Then DNA microarray technology can also be used to find out the genetic information encoded in the plant. Then what are the challenges and conservation technology? So due to increasing demand in herbal-based system, there's a thing that those herbal materials can be means there will be mix-ups. Okay, so in order to lower down these mix-ups or lower down these contaminations, so you have to there is a challenge and there is conservation technology also. And from due to increasing demand in the herbal-based drug system and substrate for development of other drugs, there is a heavy pressure on some selected medicinal plant due to they have a slower growth rate and they have a slower reproduction rate. Some other factors are agroclimatic specificity, lower population densities, overexploitation of the natural resources, and substantial loss in the habitat. So there is also irradical and continuous erosion of medicinal plant in the wild due to population explosion, as most of the population depends on the natural and forest product for their living. So traditional conservation strategies commonly, so this is the main strategy where traditional conservation strategy, community-based conservation and utility policies, regulation on transport and export policies, plant tissue culture, in situ and ex situ conservation, a botanical garden, seed bank, germplasm conservation can be effective to save and propagate the wild and endangered medical resources. And also other improvements are commercial improvements and IPR. So commercial improvements in tissue culture technique can also be possible by automation and robotization. Tissue culture in the liquid medium and use of bioreactor for the propagation of different or scaling up of their plant material can also be used for metabolite production. So opportunities and challenges can be globalization, can be patents or intellectual property right and biopiracy issues need to be approached, need to be a modernized approach in the line of development of information, machine learning, database system, on-site and off-site information, system screening and measurement. So this is what my concept on conservation and some of the concepts for improvisation of the herbal system. Thank you.
Okay, thank you, Mr. Debasish Sahoo, for this new approach to drug manufacturing and all the big drug market and the possibilities which are ahead.